right at this exact second, as you listen to the sound of my voice, something pretty incredible is happening inside your body.
Oh, absolutely. It's one of those biological miracles we usually just, take entirely for granted.
Yeah, exactly. I mean, the sound waves are vibrating in your ear right now, and your nervous tissue is receiving that auditory stimulus.
Right, and it's translating that into an electrical signal and transmitting that information into your brain to integrate a conscious thought.
All in literally milliseconds. It's just wild. So today, we're taking a deep dive into the microscopic machinery making that happen, which is your nervous tissue.
Yeah, the mission today is to really look under the hood of one of the body's four primary tissue types. We want to understand the biology of how we actually sense our environment.
And how we coordinate our responses to it. And to do that, we have to look at the two main characters of this story, which are neurons and glial cells.
Which presents a really fascinating contrast, honestly, because, neurons are the absolute superstars of biology.
Right, they get all the glory.
Exactly. They are the electrically excitable cells that carry the signals. But glial cells, or neuralia, they spent a very long time being totally dismissed.
Yeah, reading through our sources for this deep dive, it's kind of crazy. They were essentially viewed as biological packing peanuts.
Just this underappreciated support staff holding the important cells in place.
Right. And it becomes clear that for over a century, science completely misunderstood one of these cell groups. It actually led to one of neuroscience's biggest historical blunders.
It really did. But to unpack how these cells actually work together to, create human consciousness, we first have to appreciate how incredibly difficult it was to just see them.
So we have to step back a bit, right?
Yeah.
To this massive scientific feud in the late 19th century.
Yeah, exactly. At the time, microscopes were improving, but nervous tissue just looked like a dense, tangled, opaque mess.
Like a bowl of spaghetti where you can't tell where one noodle ends.
That is a perfect way to describe it. Researchers just couldn't tell where one nerve cell ended and another began. But then in 1873, an Italian physician named Camillo Golgi changed everything.
He invented this revolutionary staining technique, right, using silver chromate.
Yes, he called it la reazione nera, or the black reaction. And the absolute genius of this stain is that it didn't color every single cell.
Because I guess if it had, it would still just be a tangled black blob.
Exactly. Instead, it randomly crystallized inside only about 1 or 2% of the cells in a sample.
Oh, wow. So just a tiny fraction.
Right. So suddenly, against this pale yellow background, scientists could look through a microscope and see a few isolated nerve cells in stunning detail, completely black right down to their tiny branching processes.
But, seeing the structure of a cell and actually understanding how it functions are two very different things.
Oh, definitely. And that's where the drama starts. Golgi looked at his own stained tissue and proposed what he called reticular theory.
Right. He believed the nervous system was essentially a circulatory system, but for electrical signals. Like, he thought neurons fused together into one continuous interconnected web.
Yeah, he visualized a continuous physical network. But then in the 1880s and 90s, this Spanish neuroanatomist named Santiago Ramón y Cajal enters the picture.
And he basically uses his rival's own tool against him, right?
He absolutely does. Cajal takes Golgi's exact same staining method, the black reaction, applies it to tissue from the cerebellum and the retina, and reaches the exact opposite conclusion.
He argued for the neuron doctrine, right? Insisting these were distinct individual cells that didn't fuse together at all.
Right. He proposed they communicate by sending signals across tiny physical gaps.
So to use a modern analogy, it's like Golgi thought the brain was like a city's continuous plumbing system. Like all the pipes are physically welded together and the water just flows everywhere continuously.
That's a great way to picture it, yeah.
But Cajal realized it wasn't a continuous pipe. It was more like two people standing on opposite sides of a river throwing messages in bottles to each other.
Exactly. The river represents the physical gap, and the bottle is the chemical message crossing that gap. And ultimately, Cajal won that argument.
The neuron doctrine became the entire foundation of modern neuroscience.
It did. And decades later, in the 1950s, the invention of electron microscopes finally provided the visual proof of that river. The synaptic cleft, that actual physical space between neurons.
Which leads to this remarkably awkward moment in scientific history. Because before electron microscopes existed, Golgi and Kachal were jointly awarded the 1906 Nobel Prize in Physiology or Medicine.
Yeah, just imagine that. You have two joint winners sharing a stage who fundamentally disagree on the core architecture of the brain.
Didn't Golgi actually use his own Nobel lecture to complain about Kachal's theory?
He did. He bitterly complained about it during his speech. The tension must have been just palpable in that room.
I can't even imagine. But the sources point out Golgi wasn't 100% wrong, right?
Right, as a historical footnote, he wasn't totally wrong. We know today that electrical synapses or gap junctions actually do exist in certain places.
So they allow for a direct, fused connection between some neurons.
Yeah, so his continuous web idea does happen in rare instances. But Kajal was right about the vast majority of nervous system communication. It happens via chemical synapses across a genuine physical gap.
Okay, so now that we understand the brain is full of individual cells throwing chemical bottles across a river, we kind of need to talk about how the message gets to the riverbank in the first place.
Right, like how do neurons send electrical shouts so incredibly fast from our heads to our toes?
Exactly. To answer that, we have to look at the architecture of the neuron itself and the electrochemical physics driving it.
Yeah, you can think of a neuron as a one-way street for signal flow. It basically starts at the dendrites.
Those are the branching extensions that receive incoming chemical signals from other neurons.
Yes, like the branches of a tree reaching out to catch falling leaves. Those dendrites feed into the cell body, or the soma.
And that's where the nucleus lives, and it integrates all those incoming signals.
Exactly. And if the signal is strong enough, it triggers an electrical impulse down the axon.
Which is that single long projection that conducts the signal away from the cell body.
Right, toward the next
Wow.
cell. And it ends at the axon terminals, which release the chemical neurotransmitters.
But the sources mentioned a crucial mechanism here. This electrical spark traveling down the axon isn't like, electricity flowing through a copper wire in your house.
No, not at all. It's a biological wet medium. So it relies heavily on charged particles, specifically sodium and potassium ions.
So a resting neuron is basically like a tiny localized battery.
That's a perfect analogy. The inside of the cell is negatively charged compared to the outside. So when a neuron fires, voltage-gated channels in the cell membrane suddenly open up.
And positively charged sodium ions just rush into the cell.
Yeah, flipping the electrical charge in that specific spot, which creates a total domino effect.
Right, that localized flip in charge causes the next set of gates down the line to open, and then the next, and the next.
And that wave of rushing ions is what we call the action potential.
Okay, I want to clarify a massive vocabulary mix-up that I see literally everywhere. People use the words nerve and neuron interchangeably.
Yeah, all the time. But medically, they are entirely different structures.
Right, a neuron is a single microscopic cell. But a nerve is a macroscopic anatomical structure found only in the peripheral nervous system.
Exactly. If you picture a neuron as a single, delicate fiber optic thread, a nerve is the thick, heavy-duty cable that bundles thousands of those individual threads together.
Wrapped up in protective connective tissue. Okay, so we have this delicate action potential, this wave of rushing ions traveling down this single axon thread.
But just like a real electrical wire, it needs insulation so the signal doesn't just degrade and fizzle out.
Enter the glial cells.
Yes. Two specific types of glia handle this insulation. You have oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system.
And their entire job is to wrap axons in myelin, which is this dense, fatty substance.
Right, and the structural difference between these two is actually fascinating.
Yeah, the sources said an oligodendrocyte in your brain acts kind of like an octopus.
Right, it reaches out multiple tentacles to wrap myelin around several different axons at the exact same time.
But a Schwann cell in your arm just wraps its entire self around a single segment of one axon, sort of like a jelly roll.
Exactly like a jelly roll, and the end result is a tightly insulated wire. But this myelin is really the secret to human processing speed.
Because the myelin sheath isn't actually continuous, is it?
No, it's interrupted at regular microscopic intervals by tiny uninsulated gaps called the nodes of Ranvier, named after the French histologist who described them.
So because the myelin insulates the axon so tightly, those voltage-gated ion channels we just talked about can't open where the fat is wrapping the cell.
Right, the ions can only cross the membrane at those exposed nodes of Ranvier.
Which means instead of the action potential plodding along every single millimeter of the axon in a slow domino effect, the electrical signal literally jumps from node to node to node.
Yes. This is called saltatory conduction from the Latin word saltare, meaning to leap.
And that saves massive amounts of energy and drastically increases the speed of the signal. The numbers in the source material are actually staggering.
They really are. An unmyelinated axon conducts signals at about 0.5 meter to maybe 2 meters per second.
But a large myelinated axon can conduct signals up to 120 meters per second. That's insane.
It's the difference between a signal taking 0.5 second to travel the length of your leg versus just a few milliseconds, which is why when you touch a hot stove, your reflex withdrawal happens before you even consciously realize you're burning yourself.
Okay, but let me push back on the biology here for a second. If myelin makes everything lightning fast, and fast reactions equal survival, why aren't all our axons myelinated?
That is a great question.
Like, why would evolutionary biology ever design a slow signal?
Well, biology is incredibly frugal with space and energy. Myelin takes up a lot of physical space and requires vast amounts of metabolic energy to maintain.
Okay. That makes sense.
And honestly, not every bodily function requires a millisecond response. Digestion, for instance, can happen slowly. But if we look at a really tangible example, think about how you process pain.
Right, like if I stub my toe, I definitely want to know about it immediately.
And you do thanks to fast myelinated sensory fibers called A-delta fibers. When you stub your toe, that sharp, instantaneous jolt of pain travels on those A-delta fibers.
It's the immediate warning to move your foot.
Exactly. But a few seconds later, that sharp pain is replaced by a lingering, dull ache that might throb for, an hour.
Oh, wait, that dull ache is traveling on a completely different pathway.
It is. It travels on small diameter, unmyelinated fibers called C fibers.
No.
Yeah. Because they lack myelin, the dominoes fall slowly. So the fast fiber gives you the urgent warning to stop the damage, and the slow fiber provides the lingering reminder to favor the foot and protect the injury while it heals.
That is amazing. I will honestly never think about stubbing my toe the same way again.
Right. It's brilliant engineering.
The sources also point to the knee-jerk reflex as the ultimate illustration of this myelinated speed.
Oh, the patellar reflex. It's a classic demonstration of a beautifully simple neural circuit.
So a doctor taps your knee tendon, stretching a sensory receptor in the muscle.
Right, and that myelinated sensory neuron shoots a signal directly into your spinal cord, forms a single synapse, a monosynaptic loop, straight to a motor neuron.
Which then fires an action potential right back out to your quadriceps muscle, causing it to contract. So your leg just kicks out,
Yeah.
and the brain isn't even involved in the decision.
Not at all. The entire loop takes roughly 20 to 30 milliseconds.
That's incredibly fast.
It perfectly illustrates both the sheer speed of saltatory conduction and how the nervous system utilizes hierarchical processing. The spinal cord just handles the immediate physical reflex without waiting for the brain's permission.
Okay, so we have spent a lot of time glorifying the fast-firing neurons. But what about the glial cells making that myelin?
Yeah, let's talk about them. Because our sources point out that for roughly a century, science treated them with outright disrespect.
They really did. They suffered from a terrible naming convention, didn't they?
They did. In 1856, the German pathologist Rudolf Virchow, who is actually famous for founding cellular pathology, was examining brain tissue.
And he saw all this material packed between the neurons and basically just assumed it was passive, non-cellular structural support.
Right, so he named it neuroglia, which literally translates to nerve cement or nerve glue.
Textbooks literally treated them like biological packing peanuts, just this inert material keeping the important thinking neurons from sloshing around.
Yeah, and that assumption led to one of the most widely repeated myths in textbook history, the quote-unquote fact that glial cells outnumber neurons 10 to 1 in the human brain.
Right. If you look at older biology textbooks, that 10 to 1 ratio is stated as an absolute law.
It was everywhere.
But let me ask, if scientists are supposedly rigorous, how did a completely fabricated number end up as an accepted fact for decades? Didn't anyone just, count the cells?
Well, counting brain cells is a logistical nightmare. Different regions of the brain have wildly different cellular densities.
Right, like the cerebral cortex is fluffy and spread out, while the cerebellum at the back of the head is super densely packed.
Exactly. So historically, researchers would count a tiny slice of one specific brain region, like a piece of the cortex, see a high concentration of glia, and just extrapolate that assumption across the entire mass of the brain.
That seems like a huge leap.
It was. And it wasn't until 2009 that a Brazilian neuroscientist named Susana Herculano Husel finally figured out how to do it accurately.
She invented a technique called the isotropic fractionator, right? Which, honestly, reading the methodology is a very clinical term for what is essentially brain soup.
Yes, brain soup. It sounds chaotic, but it completely solved the density problem.
She basically dissolved human brain tissue using a detergent that destroyed the cell membranes but left the cell nuclei intact.
Right. She turned the brain into a homogeneous liquid soup of free-floating nuclei. Because they were now evenly distributed in a liquid rather than clumped in tissue, she could take a small sample, count the nuclei, and get a highly accurate whole brain count.
And her result completely dismantled the 10-to-1 myth. It turns out the ratio is basically 1-to-1.
Yeah, the human brain has about 86 billion neurons and roughly 86 billion glial cells.
It serves as a monumental cautionary tale in neuroscience about repeating unverified assumptions.
Absolutely. And recognizing their sheer numbers forced a major paradigm shift. Modern science now realizes that glia are equal partners in how the brain computes information.
They are heavily involved in active processing. Like, let's look at astrocytes, the star-shaped glial cells in the central nervous system.
Yeah, they do provide structural support, but they also physically form the blood-brain barrier by wrapping their tiny feet around our capillaries.
So they act as the gatekeepers, determining what nutrients from the blood can enter the brain tissue and blocking dangerous toxins.
Exactly. But more importantly, astrocytes actively regulate the chemical environment around neurons. They mop up excess potassium ions and stray neurotransmitters to keep the synaptic communication channels clean.
Which is huge.
Yeah.
But if they are constantly monitoring the synapses, it begs the question, are they actually processing information themselves?
And the sources confirm they absolutely are. Astrocytes participate in the tripartite synapse.
So it's not just two neurons throwing a chemical bottle across a river anymore.
Right. There's a third party, the astrocyte eavesdropping on the conversation and releasing its own chemicals to modulate the signal.
That is wild.
And even more astonishing, astrocytes communicate with each other. They don't use rapid electrical action potentials like neurons. They communicate using slow, sweeping calcium waves that ripple through their interconnected networks.
So they basically have their own entirely separate chemical communication network layered right on top of the electrical one.
Exactly. And the brain's internal ecosystem requires even more specialized glia because of that strict blood-brain barrier we mentioned.
Right, because normal white blood cells, like the body's standard immune system, usually aren't allowed inside the brain.
Right, so the brain needs its own private security force. And that role is filled by microglia.
They are the immune sentinels of the central nervous system, constantly patrolling the tissue, looking for pathogens and scavenging dead cells or debris.
You also have ependymal cells, which line the brain's ventricles and use tiny hair-like cilia to circulate cerebrospinal fluid.
And then in the peripheral nervous system, alongside the myelin-making Schwann cells, you have satellite cells, which surround and cushion the peripheral neuron cell bodies.
It's an incredibly intricate support system, and it is absolutely vital, which becomes terrifyingly clear when you look at what happens when it breaks down.
Yeah, the consequences of glial failure are severe. Multiple sclerosis, or MS, is a prime example of a demyelinating disease, right?
Yes. In MS, the body's own immune system mistakenly attacks and destroys the oligodendrocytes and the myelin they produce in the central nervous system.
So it literally strips the insulation off the brain's wires, leaving behind hardened sclerotic scars.
Exactly. And without that tightly wrapped myelin, those voltage-gated ion channels we discussed earlier are exposed where they shouldn't be. The electrical charge just leaks out.
So that beautiful, fast saltatory conduction completely breaks down.
It does. The nerve signals slow to a crawl, become erratic, or just fail entirely, which manifests as vision loss, muscle weakness, and severe coordination problems.
So really vivid clinical illustration of why that quote-unquote passive glue is actually critical infrastructure.
Absolutely. And there's another vulnerability built into glia. Mature neurons generally don't divide. Once you have them, they're locked in.
Yeah.
But mature glial cells retain the ability to divide and multiply throughout your adult life. And the terrible cost of that ability is the risk of cancer.
Right, because they can proliferate. Tumors of the nervous system overwhelmingly arise from glia not neurons.
Yes, like glioblastoma multiforme. It's a cancer of these astrocyte lineage cells, and it's highly aggressive. Just as a factual historical reference, taking no political stance, this is the exact same disease that claimed the lives of U.S. Senators Ted Kennedy and John McCain.
It's a very sobering reality, and the protective nature of glia can also backfire in the event of physical trauma, like a spinal cord injury.
Yeah, if you cleanly cut a peripheral nerve in your arm, it might actually slowly regenerate and heal. But if you sever axons in the central nervous system, they cannot regrow.
Why is there such a difference?
Well, in the peripheral system, Schwann cells actively support and guide axon regrowth. They literally form a tunnel for the severed nerve to grow through. But in the central nervous system, the environment is actively hostile to regeneration.
Because of the astrocytes.
Yes. When there's a spinal cord injury, astrocytes rush to the site and proliferate densely around the wound, forming a glial scar.
So they're trying to stop the bleeding and wall off the infection, which, makes sense in the short term.
Short term, it's a brilliant survival mechanism, but long term, that glial scar forms a dense physical and chemical barrier that permanently blocks the severed axons from ever growing back and reconnecting.
Wow. So a major focus of modern neurobiology must be trying to figure out how to get the protective benefit of the astrocyte response without the permanent cost of paralysis.
Exactly. It's a huge area of research right now.
We have covered a tremendous amount of ground today, moving from microscopic anatomy to whole brain pathology. But to bring all this biology directly back to your daily life, the next time your arm falls asleep and gets incredibly tingly, you now know what's happening.
Right, that's just physical compression squeezing the nerves, temporarily disrupting the flow of those sodium and potassium ions.
Or when you go to the dentist and get a shot of lidocaine, you can appreciate the exact mechanism keeping you comfortable.
Because that lidocaine is chemically designed to physically block the voltage-gated sodium channels in your neuron membranes. If the sodium can't rush in, the domino effect can't start.
So it exploits the basic electrochemical physics of nervous tissue to shut down the pain message before it even reaches the axon.
Precisely. Looking back, it's just amazing to summarize how far the science has progressed.
Yeah, we started with Golgi and Kachel in the 19th century, fiercely arguing over the physical shape of a cell, and ended up realizing that the passive glue of the brain is actually an active, dynamic partner in every single thing we do.
Overturning that 10-to-1 myth and recognizing the active role of Glia has completely reframed how we view human consciousness and brain computation.
Which leaves me with one final, truly mind-bending thought from the research for you to mull over. We spent the last century believing that everything making you was just an electrical spark jumping between neurons.
Right.
But if astrocytes, the cells we completely dismissed as packing material, are actively modulating those synapses.
And communicating with each other using their own slow, sweeping calcium waves across the brain.
Exactly. Does that mean some of our thoughts, our memories, or even our underlying moods aren't just flashing in a fast electrical circuit?
That is the new frontier.
Are those core parts of human experience actually swimming in a slower, chemical hidden web that we completely ignored for over 100 years? Think about that right now as the sound waves of my voice hit your ear and your neurons and your glia work together to process exactly what that means.